Okay, hello everyone. My name is Mojgan Rabiey. I'm very excited to be here and talking about bacterial canker in trees and how we can manage them. So we all know and appreciate that trees and forests are really important for environmental, social, and economic factors. However, there is a wide range of abiotic and biotic factors threatening our trees and what they provide for us. For example, we have pests and diseases. They are very important, and of these bacterial cankers cause problems in a range of different trees. So bacterial canker is a disease caused by bacteria that infects almost all parts of a tree. So on stems and trunk, the disease causes sunken and dead areas of bark, usually develop in spring or early summer, and they are often accompanied by vertical crackles or discharging gummy ooze. And the infected shoots will die back rapidly in spring, and on leaves, we can see a small brown spot that eventually they fall out. So our trees in the UK are vulnerable to attack by bacterial cankers. For example, we have bacterial canker of ash trees. You can see the lesions and vertical crackles on the bark and stems, branches, and twigs. Then we have bacterial canker, also is a disease of the stems and leaves of cherry trees. It causes sunken patches of dead bark and small holes in leaves. And in severe cases, it discharges huge amount of gummy ooze that looks like tree is bleeding. Also, mature oak trees are vulnerable to bacterial diseases, and the disease is characterized by stem bleeds from vertical crackles on trunk and inner bark necrosis that is caused by a polybacterium consortium in which Brenneria gibstella play important roles. Also, bleeding canker is a disease of European horse chestnut trees in the UK. It is characterized by the appearance of bleeding cankers or lesions on stems, trunks, and branches, and it can kill affected trees. My talk today will focus on bacterial canker of cherry trees that is caused by Pseudomonas stringy pathogens. So there are some control measures available to reduce the risk of disease occurrence and spread. We have building resilience, so through resistance, recovery, and adaptation. So it's all about driving long-term changes to strengthening our trees, our forests, so they would be resilient to future threats. Also, we have silviculture. It's about managing the structure, production, and ecosystem values of our trees. Is about restoring and regeneration of our trees, either naturally or by planting them. Also, we have breeding for resistance. So it's a desirable alternative method of control, but it all depends on how complex the nature of the disease is. Also, we can use a species other than the disease target, so to reduce the risk of disease occurrence and spread. However, current control measures for many of these bacterial diseases are limited or relatively ineffective. For example, sometimes the bacterial pathogen causing the disease can evolve and resist to the new control measures available. So for next few slides, I will talk about bacterial canker of cherry trees and the research we have done to control the disease. So bacterial canker is a major disease of Prunus species, including cherry, plum, or other stone fruits. It is caused by pathogens within the Pseudomonas stringy species complex, including Pseudomonas stringy pathovar stringy, or PSS, or Pseudomonas stringy pathovar monosporonorum, PSM. There are two races, race 1 and race 2. So Pseudomonas stringy is a globally important plant pathogen. It infects a wide range of important crops and it causes a huge economic loss. So as you can see here, they are able to infect throughout the year. The pathogen invades the woody tissue in winters, colonizing cambial tissues and causing canker in spring. During growing periods, bacteria enter leaves, causing the lesions on it that cause this, they drop out and produce short hole symptoms, and they cause necrotic lesions on fruits. So it is an annual problem for the global cherry fruit industry, and it's been reported to cause up to 75% loss of our cherry trees. The chemical control for this disease is currently limited to copper-based compounds. However, it's a treatment that's been restricted due to the adverse environmental effect and because it's not highly effective. Also, we have breeding for resistance. So it's an alternative method of control, but progress has been hampered by the complex nature of the disease and the need to prevent infection by 3 different clades and pathovars of Pseudomonas stringi. So another desirable alternative method of control can be use of biological control agents, like using bacteriophages. So bacteriophages or phages or bacterial eater are naturally occurring viruses that infect and kill only targeted bacteria. They are a highly desirable option for management of bacterial diseases because they are extremely selective to the target bacterium and they will not adversely affect other microorganisms, and because one phage can destroy a bacterial cell and in the process it produces hundreds of other phage particles that infects neighboring bacterial cells. Also, they can be used in an existing program as a resistant management tool. So in our ViroPlant funded project, we used a bacteriophage to control bacterial canker of cherry trees. So we collected huge number of samples from cherry orchards in the UK, from leaves, barks, and soils of cherries. And we took the samples to the lab and we isolated and purified 70 phages. And we characterize them. For example, we visualize them under their morphology under electron microscopy. We whole genome sequence them to find out they don't have any toxin genes that limit their use. We tested the host range assay to see which pathovars and strains of Pseudomonas stringi they target, and to find that they don't have any effect on other beneficial microorganisms. Then we sub-selected 13 phages and we tested the efficacy and virulence in the lab, and then we tested the efficacy in planta. So we tested the efficacy of the phages on cherry leaves and twigs individually or in cocktails. So it means mixture of phages. You can see cocktail 13, mixture of 13 phages, and cocktail 7 is a mixture of 7 phages. So we first sprayed the leaves with PSS or PSM1 or PSM2. Then we sprayed them with phage individually or in cocktail. Then we collected samples every week for 5 weeks and counted the colony-forming unit of bacterial population. So as you can see in the graph here, PSS population alone increased throughout the experiment. However, when we applied cocktail of phages, the population reduced to almost 0, so it's very effective. However, the fluctuation you see here, it might be due to bacteria resisting to phage or due to the use of multiple phage causes and antagonism effect between phages. So how we can apply phages to the fields to hundreds and thousands of large trees? So there are currently a few phage products available in the USA, like AgriPhage fire blight against fire blight of apples and pears caused by Erwinia amylivora. We have AgriPhage citrus canker against Xanthomonas citri. They are being sprayed on leaves. So they are being sprayed early season as a preventative measure, or mid-season when the symptoms of disease appear, or late season when the severity of disease is high. Also, we can reduce bacterial population resistant to phage by using phage cocktail. Also, we can use the lower dose of phages in cocktails. The lower the dose of phage, the lower the rate of bacteria resisting. Also, we can do evolutionary experiments to find evolved phages that can target resistant bacteria. So we are currently working on a BBSRC funded project, working on bacterial canker of cherry trees. So what we know about this disease is there's significant variation in resistance to canker within cherry cultivars and also between genotypes of wild cherries. Also, what we know that there are epiphytic population of Pseudomonas stringi living on the surface of non-diseased cherry leaves. And also we know that the epiphytic population, they have genes predicted to have a key role in host adaptation and pathogenicity to Pronunciate species. But what we don't know is whether this epiphytic population of Pseudomonas stringi are similar between cultivated and wild cherries. Also, what we don't know if whether this epiphytic population have pathogenic potential. So we want to study the mechanism of host range expansion of Pseudomonas stringis to explore the genetic factors that may affect the pathogenicity, the virulence, and what leads to new disease outbreaks. So we are identifying factors that regulate pathogen ability to persist on trees. So thank you all for listening. And if you have any questions, I'm happy to answer. Thank you.
Thanks very much. That's a great talk.